great to welcome back Jan Frankowski, who gave a beautiful talk back in the spring last year and is back now to moderate the seminar. And so I'll pass it on to you. Thanks.
All right. Today, it's my pleasure to introduce Dr. Bret Smith, Professor of Physiology and Chair of the Department of Neuroscience at the University of Kentucky. Dr. Smith received his PhD from the University of Tennessee Health Science Center, where he studied the neuromodulatory effects of histamine in the hypothalamus under the mentorship of Dr. William Armstrong. Dr. Smith went on to complete his postdoctoral training at Colorado State University under the mentorship of Dr. Ed Dudek, where he studied circuit reorganization in the basolateral amygdala and hippocampus in epileptic animals. Since Dr. Smith started his lab at Tulane University in 1999, he's published over 60 peer-reviewed publications focused on understanding the reorganization of brain circuits in post-traumatic epilepsy and temporal lobe epilepsy, all the while continuing his work on understanding the organization and function of autonomic circuits. Since Dr. Smith joined the faculty at the University of Kentucky in 2006, he served as Director of Graduate Studies, Director of the Epilepsy Center, Director of the T32 Program in Integrative Physiology, and Chair of the Department of Neuroscience. In addition to his academic and administrative roles, he's trained 28 postdocs and graduate students, including my own graduate advisor, Bobby Hunt. So I guess that would make him my academic grant mentor. So with that, I'd like to give a warm welcome to Dr. Smith, who today will be talking about modeling cellular mechanisms of post-traumatic epileptogenesis.
Thank you, Jan, and thanks Adam and David for organizing all this. Well, let's see. Hope everybody can see that, and I'll just my pointer along the way. The first thing I want to do is get right to the acknowledgements, because there have been a bunch of people working on this, and I don't want to get to the end and forget anybody, and I probably have forgotten somebody as it is. But the list on the side is the people that have contributed directly to the work that's gone into this talk. And especially, you'll notice a couple of them will come up over and over. Corey Butler, who's a postdoc in Oregon now. Bobby Hunt, obviously, is there at Irvine. Sang-Hoon Lee and Ginny Kang are with me now. And Ryan Cloyd is a MD PhD student in the lab now working on this project. So I want to thank everybody on that list. Steve Sheff got us started in this, and Kathy Satman is a current collaborator on some of the work that's funded right now. And thanks, of course, to the people who have, and the groups that have funded this over the last however many years. I know this group is a TBI group, so I'm going to start off by telling you about epilepsy, because the talk is about modeling post-traumatic epilepsy, and in order to do that, you sort of have to merge two fields. And I know a lot of people, there are probably a bunch of people in the audience who have studied some form of epilepsy or another, and because people do that to sort of learn about neuroscience. But in case you haven't, I want to get into this just a little bit, because it actually forms the way we think about epilepsy in general, but especially post-traumatic epilepsy. So this is sort of a basic slide just to separate in your minds the idea between a seizure, which is just an event, a clinical manifestation as it's defined of abnormal, it's excessive and coordinated excitation of populations of cortical neurons, versus epilepsy, which is nothing more than a tendency for this to happen more often. So seizures are sort of a symptom of an underlying disorder in the brain that makes the brain more likely to go into this state of hyperexcitation. And so specifically, I'll be talking about acquired epilepsies. There are genetic forms, and there may or may not be a genetic or hereditary component to those. But acquired epilepsies, symptomatic epilepsies, are then where everything is fine, and then at some point something happens, and a person starts, usually in young adulthood, starts to have recurring seizures. And so brain injury is probably the #1 predecessor of that kind of epilepsy, which makes it important in this field. There have been a number of hypotheses about how the brain becomes epileptic, and basically it boils down to this. There's either too little inhibition, and this can be at the cellular level, talking about ionic currents going across the membrane of neurons, or it can also include neurotransmitters, so death of GABA neurons or reduction of inhibitory GABA circuitry. But there can also be too much excitation. And so it's probably a combination of both of these that cause the epileptic phenotype, and it's chronic. And so what it does is, and I'm going to show this kind of a dumb diagram at the bottom here, where we've got this sort of balance in the brain and the circuitry. This is not what your brain looks like, but this is how I think of it, between GABA and glutamate, and it might be tilted a little bit to the left or right, but they're basically in balance. And so most of the time, our brains are okay. Something happens eventually to either reduce the inhibition or increase the excitation to tip that balance. And that happens occasionally. And so one of the key things to remember is that people with epilepsy are not having seizures all the time. Most of their brain function is just fine most of the time, with this sporadic, episodic seizure events superimposed on top of normal behavior. And of course, there are some other comorbidities that go along with that can reduce overall function behaviorally, cognitively, in many of the same ways that people that study TBI study them. But for the most part, people are not having seizures all the time. And that's important to remember because there are some components of this that are chronic and some that are episodic, and we'll get into that. I've been working for about 30 years now on developing epilepsy models in animals. And just to remind you that, you know, I don't know, there's a quote out there somewhere, and I can't remember who said it, and somebody's going to remind me that all models are bad or imperfect, but some are useful. And so what we need to do is define the utility of our models. And so I'm not going to talk to you about the translational aspects of any of these models, because as we know, as people who study TBI or epilepsy know, that it's really hard to model an entire disease in a way that allows us to study aspects of it in a truly translational way. And so what I've done is I've tried to refine this so that when we're talking about models, we're talking about using a model of a disease to study components of that disease. So cellular correlates, for example, of epilepsy. And they include some loss of cells, axon sprouting, synaptic reorganization, local excitability changes, and then also what I won't talk about much is receptor and channel redistribution. In general, the way we think of this for acquired epilepsies is something happens, an injury. I'll talk a little bit about some of the models we've used in the past using status epilepticus, then also TBI. Something happens, and then there's a period where everything seems okay-ish. And that can be a period lasting weeks or even in some cases years. But during that time, something's happening in the brain to change the way it functions, to change the way it's put together, so that eventually you develop epilepsy where you've got this permanent, or at least we think permanent structural functional change in the brain that makes it more susceptible to having seizures. We like to think about brain circuits when we do these experiments. And so this is sort of a circuit way of thinking about that. Here's a basic circuit. There's a principal cell that kind of looks like a granule cell, dentate granule cell. There are a bunch of them. They're lined up in cortical areas in layers. And then there are these local interneurons that control their activity very tightly. And in general, the interneurons, the inhibition of these circuits dominates the system, unless there's something important for this neuron to do. After an insult, and for the following, especially days and sometimes years, there are a number of things that happen. And one of the more important ones is probably a loss of some of this inhibition. But there's also a growth of axons and a synaptic reorganization such that these sort of sparse connections between the principal cells in some areas of the brain anyway can be amplified. And so now we get this circuit change over time where we have new connections between the excitatory neurons and a loss of input from some of the inhibitory neurons that tips that balance. And this is the way sort of the field has modeled these circuits for a very long time. I've seen diagrams like this going back to the early 80s for how they think circuits are modified. And so I'm going to sort of press this a little bit forward to you to tell you about the things we kind of study and then back it off and look at what happens in TBI. So I'm going to start with a more classical model of temporal lobe epilepsy where we use pilocarpine, which is a muscarinic agonist. after we block peripheral muscarinic receptors, so we don't have autonomic effects, you inject pilocarpine, and that the mouse or the rat or whatever rodent you're looking at or animal will go into what's called status epilepticus, which is a series of seizures and these seizures can last, and they're more or less continuous in these animals for a couple of hours. So a couple hours of seizure activity, and then they're fine. The animals are fine, they wake up, they're a little woozy for a couple of days, and then they go around doing their mouse thing for a period of a few weeks. That's the time, the latency period. And then they start having spontaneous seizures, spontaneous recurrent seizures, some people say, that's accompanied by, for example, sprouting of axons in the hippocampus, mossy fiber sprouting. The seizures are stereotyped in rodents. And actually Bobby Hunt took these pictures and I show them reluctantly, but they're really good shots of what we call a category 2 seizure. Category 3, these become generalized. Category 4, they're lifting both arms. You can see his forelimb is up, tail is stiff. Category 5 is a loss of posture. These were defined back in the 1970s by Racine. And the mice basically follow this same stereotyped seizure activity for temporal lobe epilepsy. There was a controversy sort of in the late 90s as the neuroscience field started shifting from rats to mice because we started making transgenic mice, and there were a lot of studies we could do, but some of the status epilepticus models, the more popular ones, just didn't seem to work in all strains of mice. And if you work with mice, you know that some strains are different than others. And so we managed to get this pilocarpine model transferred from rats to mice fairly effectively, and it turned out that a couple of different strains, if we could induce status epilepticus successfully in the mouse, most of them, and these numbers, this is from a paper in 2002, but these numbers are significant. Most of these mice, if they undergo status and survive, they become epileptic a few weeks later. And so we focused our studies on, since it's temporal lobe epilepsy, on what happens in the temporal lobe. And this on the left is a diagram of where the hippocampus is in a human skull. You can see where it is, sort of in the middle of the brain there. This is just an image. A mouse brain is significantly smaller than a human. The hippocampus is, of course, relatively larger in a mouse than it is in a human. And a lot of the studies we do will involve taking out the hippocampus and making slices and keeping them alive and recording from neurons in that structure. The reason we choose that is based essentially on work from resected human tissue. So a lot of times if their seizures are intractable and they're in the right category, they can have a hippocampal resection to try to treat their seizures, and it's fairly effective. But what they saw when they stained those human samples years ago, decades ago, was that the axons of the granule cells, which are shown here, this dark stain, this is the dentate gyrus, and those are dentate granule cells, and they project their axons to CA3, so basic neuroanatomy, and they stain. They have zinc, and so we can chelate the zinc and stain them, and that's what they look like in a normal animal. If you blow up this section, you'll see that the granule cells are lined up, and the axons are where they're supposed to be in the hilus, heading towards CA3. And I clicked my screen. But in an animal that has developed epilepsy, and this is about 2 months after going through status, you can see the structure's changed. And so now here they're granule cells, but you'll see this dense band in the intermolecular layer of the dentate gyrus where it's not supposed to be. In other words, and you can blow it up here in D, and you can see that some of the axons are traversing through the granule cell layer, and they're actually making synapses out here with other granule cells. And so you have a structural change that underlies a functional excitability change, we think, in the dentate granule cell layer that just doesn't exist in a normal animal. This is important because for many years, anti-epileptic drugs were all aimed at stopping seizures in a normal brain. And it wasn't until about 20 years ago that drug companies finally, with some pressure from NIH and the American Epilepsy Society, finally started really seriously looking at the effectiveness of those drugs in this remodeled circuit, which seems to be intuitive, but wasn't at the time for some reason. So what happens there? If we record from those cells, this is a diagram of the hippocampus. There's the granule cell layer. You can see one granule cell with its dendrites out here in the inner and outer molecular layers, its axon heading towards CA3 like it's supposed to. You can stimulate those axons and cause an antidromic action potential in this cell. Since they're all lined up, it's easy to record field potentials or individual neurons, and that's what this is. In our experiments, we almost always cut off the main inputs to the hippocampus, so it's kind of isolated from any kind of reverberating back and forth. And so it isolates the circuit. If we record in a condition where we block synaptic inhibition and we jazz things up, any excitatory connectivity that is there using magnesium-free medium, sort of exposing the NMDA receptors, and we do that antidromic stimulation in a normal mouse, you get an antidromic population spike, and that's what this is. That's what you see with all the inhibition blocked, or the GABA-A inhibition blocked anyway, and NMDA receptors exposed by lowering magnesium. If you do the same thing in a mouse that has been treated with pilocarpine, undergone status and survived, and then look about 6 weeks later after it's developed seizures, sometimes you see this sort of longer lasting population activity that suggests activity of a population, a group of neurons that's beyond the single action potential. And occasionally you see this, more than occasionally, you'll see this lasting a very long time. And this, if you look at the time scale on this seizure-like event, as we sometimes call it, can last a minute and a half or even 2 minutes. These are just blow-ups of that. You can see the population spike under the first arrow, followed by this sort of variable delay to this population depolarization, which suggests that there's sort of a reverberating circuit underlying this. And so what we think has happened is that these granule cells now have new axons. They're sprouting new axons, and they're making connections with other granule cells, which tips the balance of that equation. Okay, that's fine and good. Not very many people develop epilepsy after undergoing status, especially from injection of a substance like pilocarpine. It can happen under different circumstances, but the field of epilepsy is kind of known for quite a while that the #1 predisposing factor for developing epilepsy, temporal lobe epilepsy or focal epilepsy, is probably a brain injury. The issue, as most of you know, is we as scientists get into our fields and we study epilepsy, and the brain injury people study brain injury. And it's very difficult to actually find people who are willing to take 2 different fields and put them together in one field. And so, you know, it's hard to even get an epilepsy researcher to understand what traumatic brain actually is. And so we say it's a mechanical force that disrupts normal brain function. That's a pretty simple explanation. But even that is sort of, we're not trained to think like that. So post-traumatic epilepsy is actually an epilepsy like the kind that I just showed you, but it's induced by brain trauma. And you develop unprovoked seizures that can take weeks to years even. And there have been studies that suggest that brain injuries occurring during the Vietnam War could take even 10 or 15 years for the patients to develop epilepsy. That's unusual. It's a little shorter than that usually. It's thought to be the cause of more than 20, it's probably closer to 30% of symptomatic epilepsies. This is a little difficult because when people develop epilepsy or post-traumatic epilepsy, maybe they're in their teens or 20s, and you know, it's a large number of them are male because men do things and hit their heads more often. And so it's hard to find people that have never had a brain injury by the time they're 20 or 22. But separating it out with lots of people, this is affecting millions of people. So this is statistical, but it looks like TBI is probably the #1 cause of symptomatic epilepsy development. It's not always temporal lobe epilepsy. Sometimes these are neocortical epilepsies, which are harder to detect. There is, in humans anyway, there's a correlation between the risk, the severity of the TBI and the risk of post-traumatic epilepsy, with, you know, maybe half or even more of people that survive a penetrating brain injury will develop temporal lobe epilepsy within 5 years, but that's more rare and often the least of their problems. And so what we really are looking at is these milder to moderate brain injuries that still increase the possibility of developing temporal lobe epilepsy dramatically. Normally, among the entire population, about 1% have epilepsy. And so if you're talking about 4.2% of those that have undergone a TBI, you're talking about a 4-fold or 400% increase in the probability. So it's a lot. And this is something that is not so easy to find in the literature, but actually exists. And that is that Of those patients that have a brain injury that results in a brain contusion, this number jumps up to 11 to 22%. 11 if it's unilateral, 22 if it's bilateral, which it very often is, because there's often a contra-coup injury if there's a focal cortical contusion. So the risk is can be pretty great. This is a slide that Bobby Hunt put together for a review we wrote a while back, and it actually gets to some of these issues that we like to think about. So there's this primary injury that can result in acute cell death and some other things, axon injury especially, acute excitotoxicity. Then there's this period afterwards that can be hours to days later where there's hypoxia, there's some delayed cell death, apoptotic cell death, delayed necrosis, stress, inflammation, and these can cause early seizures that can actually exacerbate the primary injury. But over time, months and years later, these repair mechanisms kick in and take over. And they include neurogenesis. So there are parts of the brain that undergo continuous repair and neuron growth, including the dentate gyrus. It takes time for synapses to remodel themselves, especially if it's a morphological change. There's also angiogenesis, which we won't talk about much, but can contribute to this. And it's thought that these sort of combine to cause these late post-traumatic seizures that we define as something that occurs at least a week after the initial injury. And so I put this in here to remind myself that you and I are studying the same problems. So people that study traumatic brain injury are looking at lots of different things in terms of functional recovery and whatever else they're looking at, cognition, and people that study epilepsy are looking at some of those same things from a different perspective. And so you know this parable about the blind men who are each looking at the elephant or touching the elephant, but they're only seeing the part they study. And the trick is to try to discern what it is you're studying by looking at only one part and not having to touch all the parts. It's a little bit of an odd, might seem like it's a little bit of an odd analogy, but in some ways I think it's very true that we are in this field together, and this is actually something that when we're looking at things like traumatic brain injury, we need to start looking at things more globally, and that's kind of what started me in this whole field. There's a long story that goes with that, but I won't tell it. So when we model post-traumatic hyperexcitability or post-traumatic epilepsy, there are lots of different ways to do this. Of course, I talked about penetrating brain injuries. These are models that are out there. There are non-penetrating injuries, which are much more common, that result in either focus or diffuse, and sometimes they can be both. And so lateral fluid percussion injury is used very often, for example, and that's a diffuse injury. The people that turn that into a post-traumatic epilepsy study tend to hit them with a fairly high pressure. So they end up often getting a focal injury on top of that. And the people that do those studies are now acknowledging that there's a focus to it as well. There are advantages and disadvantages to all of these. And so for the fluid percussion, there's a fairly high mortality in the animal models. There's some variability. It's not so easy in mice. Not that many people are successful with it. And among the more difficult aspects of modeling these things in this particular injury model is that to get epilepsy to emerge, even in rats, can take a year. And so it's been fairly well defined. Asla Pekkanen has done this for years now, and she says in her rats, after about 6 months, about 50% of them are having seizures, and the percentage increases over time so that by about a year, most of them are having seizures, and that actually has some great advantages. But if you have a trainee in the lab, a postdoc, who's going to wait a year before they can do their study, it's not that great, and you have to wait a long time for these things to happen. Weight drop has been used in a few studies. There are some issues with that as well. What we settled on was the controlled cortical impact. And so there's a low mortality. It's a fairly consistent injury because we can control the depth and the speed of the impactor. And then the seizures start to occur in about 2 months in about 40% to 50% of the animals. So that's, to me, seems like it's a fairly good model to study some of the things some of the changes we want to look at. Here's a diagram of the impactor. There's the mouse, make a little hole. This impactor touches the dura and pushes on it, and ostensibly, we hope, doesn't break the dura, but causes this contusion injury. And it happens if we do it over the frontal parietal lobe, it hits the hippocampus or comes close to it. if you fix a mouse brain, take it out, you get this kind of a big hole, and this is bigger than what you and I would probably sustain and survive, but mice do fairly well. So we'll do the injury, we'll wait a couple of months, monitor for seizures. That's important because if it's epilepsy, you've got to know whether they're having spontaneous seizures. And then we start looking at the electrophysiology and the histology. So there's another good long story that went into this, but when Bobby Hunt was in the lab, he decided to try this. And he found that a fairly large percentage of his mice, after a couple, 3 months, started having seizures. And this actually became a very predictable model of epilepsy with spontaneous recurring seizures developing after a brain injury. That I was very happy that he found this and took the time to do this, because not everybody would do that. He slept in the lab, by the way, while he was doing these studies, and as all good grad students should. But he was very careful, and he ended up creating the first truly viable model, I think, of post-traumatic epileptogenesis. So what did he do? He repeated those studies like what we did in other temporal lobe epilepsy models, and did that anadromic stimulation, and saw that in controls, or if there was an injury but no axon sprouting, which you could detect post hoc, they look kind of normal. But in animals that had developed spontaneous seizures, and then post hoc, he could do the staining and find that there was mossy fiber sprouting in the intermolecular layer. He got that same kind of response, which looked very much like what we saw in our animals with temporal lobe epilepsy induced by pilocarpine-induced status epilepticus. Another question we want to ask is, are those sprouted axons really contacting other granule cells? And we can do this by recording from a granule cell and selectively stimulating tiny areas, very focally, of the granule cell layer. And we use this caged glutamate uncaging technique to do this, where you can focus a beam of light through the actual microscope objective onto a very small area of the dentate gyrus while you're bathing it in this, it's called the caged glutamate compound. The compound is actually glutamate bound up with this molecular cage that prevents it to binding its receptors. But if you hit it with UV light, the cage breaks, and the glutamate is able to then activate receptors on any neuron that you're shining the light on. And if that neuron is connected with your recorded cell, you get this kind of response. And in this case, these downward deflections are excitatory postsynaptic currents, indicating that the light stimulus activated a glutamatergic input to the recorded cell. And so you can start mapping things out that way and actually determine that the granule cells are actually connected with each other, and it's not just a morphological change, it's a functional change that causes them to be connected. And the other thing, because it's light, you can move around the slice and focus in on different parts. These little dots are relatively about the right size of area. And so if you do look in a control mouse, and this is, again, this section has been stained post hoc from the slice in which the recording is done. He recorded using a whole cell recording here at the asterisk and stimulated at a bunch of sites. Most of the time in the control animal, he got no response. If he stimulates right on his recorded cell, you can see it depolarized it. This is an inward current, and this is what it looks like on the right in current clamps, so you can evoke action potentials in granule cells. And the reason I show this is because if you do this in an animal that has had an injury and has developed epilepsy, spontaneous seizures, you do the same kind of experiment where you stimulate at different places, here, whoops, on your recorded cell, and then in another place. And you can see that when he stimulates here, he gets inward currents, spontaneous EPSCs, evoked EPSCs in his recorded cell, which can generate action potentials. And so this pretty much definitively shows that in those animals with the mossy fiber sprouting, with the axons that have changed their direction and are headed up so they look like they might be synapsing on the dendrites of other granule cells, are in fact driving this local circuit. And so we look at that and we say, all right, well, there's a recurrent excitatory connection that's formed between granule cells after CCI. They have spontaneous seizures. There's a morphological undertone to that, the mossy fiber sprouting. It looks like the granule cells are connected to each other. Normally, they don't do that. We say that looks an awful lot like the cellular basis for what the field of studying temporal lobe epilepsy has looked at for many years. And so this should be a relevant model of post-traumatic epilepsy with predictable outcome measures. I'm going to diverge just a little bit and talk about one of the things we want to do with that. And one of the main reasons we want to study this is we would like to interfere with the process. We would like to say, let's injure the animal and give them something to prevent that cascade of events that leads to all of that circuitry change. And so for quite a while now, for probably 15 years, modulating mTOR activity has been one of those potential targets. And so these are just titles of a couple of papers. Rapamycin blocks mTOR activity. mTOR activity is increased in the hippocampus after either seizures or after brain injury. And so the thought was, if you can block it, you can block some of these changes. Corey Butler started working on this a few years ago in the lab. And then as he, as the graduate student nightmare of all times is that somebody comes out and basically publishes your study, which almost happened. It's close enough so that it was a little bit traumatic, so to speak. Didn't slow him down though. He kept doing his experiments. So the idea is that he can give the animals an mTOR inhibitor right after injury. He gave them daily, which is a chore for a couple of months, and then start and monitor seizures, and then do those same kind of ask those same kinds of questions we asked before. And I'm just going to show you just a quick recap of that. The panel on the left is basically a reorganization of what I just showed you from Bobby's paper. And when Corey repeated that, what he found was that yes, he could get a large percentage of slices with this long after discharge, after brain injury. But in the animals where he administered rapamycin at 2 different doses, he could reduce that or actually make it much more normal with a higher dose. And so it looks like the rapamycin prevented that mossy fiber sprouting. And in fact, when he this is just one part of the study, it did. The staining wasn't there. The rapamycin prevented many of these sort of cellular outcomes associated with development of epilepsy. And the problem is in our lab and then in others also, it turns out that if you stop treating the animals, after a couple of weeks, they start developing seizures anyway. So it's a great way to modify the way the disease progresses, but it's not the end-all treatment that we were hoping for and that the field is hoping for. However, it does have an effect. It does modify the epileptogenesis. It reduces mossy fiber sprouting. It changes some of the increased excitability that we see without the rapamycin. It also suppresses neurogenesis in the dentate gyrus, and that's the reason I'm showing you that, is because I'm going to get back to that in a minute. So it looks like, at least for this, the excitation part, the rapamycin seems to have fixed, at least for a while, which causes us to go back and start thinking about this again. So does the synaptic reorganization cause epilepsy? Everything is correlative right now. And unfortunately, in our field, like many fields, that's just the way it is. It's very difficult to do causation, gain of function, loss of function experiments that are that specific. And so we still have to ask ourselves, is this really the answer, this recurrent excitation hypothesis? And this is one of my favorite quotes. It's from, I was watching TV, this is a long time ago now, where I was actually watching a football game, and the announcer said, as the players on the field were leaning over, these are obviously not NFL players, but you get the idea. He said, those guys are out of breath and they need more oxygen. So they're bending over because it helps them breathe if they're closer to the grass. Every bit of that is true. I think every word in that sentence is probably true, because there's oxygen in the grass, and that is also true. Okay, so when we look at these things, we have this idea in our minds that we're testing these hypotheses, and we go at them and we say, this has to be the answer, it has to be the answer, and everything we do lines up so that this is the answer. And then we have to start questioning ourselves when we can't actually solve the disease based on the answers we're getting. I kept showing you a diagram with a teeter-totter tilted toward excitation, but there's an inhibition part too. And so I'm going to touch on some of that to get at the last part of this question. So the excitation part we've covered. Inhibition has, in the hippocampus, it's incredibly complex and interesting. There's a set of neurons in the hilus that project back to the dendrites of the granule cells. These are somatostatinergic. They're probably what's called hip cells. They cause some kind of recurrent inhibition, and they shunt out excitatory inputs to the granule cells that helps keep them quiet most of the time and responsive to only very strong inputs. So Corey followed up on some work that Bobby Hunt did in the lab where he looked at iPSCs in granule cells. And these are upward deflections or iPSCs, and you can see that there's not much difference between sham and contralateral to injury or contralateral with rapamycin. Ipsilateral to the injury, there is a little bit of a decrease in synaptic inhibition of granule cells. And what Corey found was that when he treated the animals with rapamycin, that decrease got even more profound. And so if you look down here, the frequency is at about a little over 2 hertz normally. After injury, it drops down to about 1.5 hertz. So it looks like there's some loss of inhibition, and that makes sense because a lot of these inhibitory interneurons are killed by the injury. But the rapamycin treatment actually made it worse. And so we thought, well, that's not good. It restores, it does some things with tonic inhibition that we won't cover on, but it further reduces the GABA release. It doesn't do much to cell death, and that's what this is just up here to remind me that cell death is reduced slightly in some areas of the hippocampus, not where we're recording near the epicenter, but in some areas. But overall, doesn't really do much to prevent cell death. And certainly when we focused that in on the inhibitory neurons, they were also equally susceptible. So rapamycin is not directly addressing cell death in this model. We do have this mouse where the inhibitory interneurons, or a subset of them, express GFP, and so we can target them for recordings. So these are somatostatin positive, GFP positive neurons. It's a subset of the GABAergic interneurons. And so what I'm going to show you next is recordings from these interneurons to try to see what's exactly happening with those cells in this model. And so the first thing is that when we look at the excitatory drives, so these downward deflections are EPSCs in recorded inhibitory interneurons, we can compare control and after brain injury, ipsilateral to the injury, and you can see just by this sort of low resolution trace that there's a pretty big increase in the frequency of EPSCs on these neurons. And it turns out that after rapamycin, that increase is still there, but it's less. So what it looks like is that there's less excitatory input to these cells in the hilus, these inhibitory cells. And when you look at their action potential firing, it sort of reflects that. They fire more action potentials. These are on-cell recordings of just sodium currents. But in animals that have been treated with rapamycin, it's still increased over background, over control, but it's significantly less. And if you look at this graph, if you remember the previous one, it looks a lot like it. So the action potential activity of these interneurons reflects their excitatory synaptic input change. And so we made this model and we said, well, here are interneurons. Maybe they're also getting inputs from excitatory neurons, including granule cells in the slice. And maybe that is increased after brain injury. And maybe it comes from somewhere else to actually, even though there are fewer of them, you notice that most of them over here, there are 4, and over here there's 1. So some of them have died, but maybe the surviving cells are being hyperstimulated by this excitatory circuitry to sort of compensate for the loss of those neurons. So you can do that again using the same kind of experiment as before, where we use the glutamate photolysis and stimulate granule cells, but instead of recording from other granule cells, we're going to record from these interneurons, stimulate them, and look for responses in those interneurons. And so we do that, and we say, well, this is from Corey's work, but Bobby actually did some of the earlier studies with this, showing that if you do that experiment and stimulate around the granule cell layer and record from an interneuron, you're much more likely to get responses, excitatory responses, after brain injury than you are in a control animal or contralateral to the injury. And if you treat them with rapamycin, most of that excessive input from the granule cell layer, but not all of it, most of it goes away. And that's graphed over here in B. So what that tells us is that after the injury, that axon sprouting, the mossy fiber sprouting, that we know connects to other granule cells, that we know rapamycin is preventing, is also preventing this possibly restorative or reducing the possibly restorative aspects of this. And that's fine. That is a little bit what's expected. It's disappointing, but that's maybe why rapamycin doesn't work as well as we thought. On the other hand, Corey went on and did these experiments where he stimulated in CA3 and found that CA3 also sent, after brain injury, these CA3 pyramidal cells, which normally project to CA1 and are part of this circuit that was defined 100 years ago by Cahal, are now making what we loosely term back connections to these inhibitory interneurons. And you can see that here. This is a stimulation at 0.6 and 0.7 where you get this excitatory response. And this is just an example, but you got to do this in hundreds of or dozens of cells and hundreds of stimuli. But the main take-home message is that the treatment with rapamycin did not block that. It did not undo it, and that's shown here. So unlike with granule cells, the rapamycin treatment did not undo that potentially compensatory connection. And so it's diagrammed here to the right. If you look in B, the red circles are indicating where there are new synapses, so granule cells connecting with each other. There are more granule cells connecting with hilar interneurons, and there are more CA3 pyramidal cells connecting with those interneurons after brain injury. And in rapamycin-treated mice, that's all reversed except for this input from CA3, which is maintained. So we know that mTOR does a lot of things. We know that rapamycin prevents things like cell growth, but that's still weird. How could that be that you've just got a fairly selective reduction in this circuit? It doesn't make any sense unless we start thinking about what else might be involved. And one of those is neurogenesis. I told you moments ago that rapamycin treatment prevented the excess neurogenesis that occurs after brain injury. There's been a story out there that these newborn granule cells might actually be contributing to these abnormal circuits. And so we thought maybe what's happening is that if we prevent neurogenesis, we might actually disrupt this treatment, the development of epilepsy, and that the rapamycin might actually be operating that way to prevent neurogenesis. On the other hand, the TBI field has been trying for years to find ways to upregulate neurogenesis to restore some of the functions. And so we have this dilemma. If rapamycin reduces mossy fiber sprouting, it's also going to reduce inputs to the inhibitory interneurons and may not be the best outcome that we're looking for. So we started studying this a few years ago, by getting a Gli1-Cre mouse. And so this is an inducible, Gli1 is one of these precursors that sort of early stage neurons start to make. We combine that with a mouse. We made it with a mouse that's a channel rhodopsin mouse that has an EYFP reporter. And so we end up with is mice where channel rhodopsin is expressed selectively in newborn granule cells that are born when we tell them to be born by treating them with tamoxifen. And this is just a proof of principle experiment where the animals were treated for about 4 weeks with tamoxifen. You can record from granule cells. They're EYFP positive. They look like neurons. They show action potentials. You can evoke action potentials in them by shining light. If you make the light a little stronger, you get more action potentials, and they're receiving excitatory inputs. In other words, they integrate into the circuit, which is important. So here's the experiment that we're doing right now that we're finishing up. And that is we're treating them with tamoxifen at 3 different times. One is early in life, postnatal day 8 to 12. So we treat them, and this should induce channel rhodopsin expression in a subset of neurons that are born at this time frame. By the time they get to the brain injury at P7, most of those neurons are intact and are actually probably fairly mature. And then when we look 2 months later, what we're looking at is activity that is dependent on neurons that were born early in life, but were mature at the time, or relatively mature at the time of injury, and are certainly mature by the time we do the experiment. We compare that to looking at neurons that were born just before injury or neurons that were born just after injury to see how they integrate into the circuit. And the result of this is actually a little bit of a surprise. We initially thought we'd start looking for evidence of recurrent excitation in these cells and found almost none except for with the early born neurons. But what we did find is that when we stimulate neurons that were born just before CCI, the most common response is a recurrent inhibition, which means that these neurons seem to be integrating into the circuit, and it's far more than what we see in sham animals. Even in shams that are fully mature, they seem to be preferentially activating inhibitory interneurons that then feed back onto the granule cells. Because when we record in adult granule cells that are not labeled, we get predominantly this IPS. We can show that by blocking either GABA A or ionotropic glutamate receptors. And so we know this is a polysynaptic response due to activation of newborn granule cells that feed through this circuit to cause a recurrent inhibition of other granule cells. And it's much more common for those neurons born before injury. Those neurons born after injury contribute almost nothing to the circuit. And then the early born neurons also contribute. So there might actually be some sprouting of axons. I'll go through this really quickly. The mice develop epilepsy. There are new recurrent excitatory and inhibitory circuits that come out of this that correlate with the development of epilepsy. And some of this is modified by changes in adult neurogenesis rates, especially for those neurons that are born just prior to TBI. And they actually contribute to this compensatory response. We can inhibit a lot of that with mTOR, but mainly we think through the neurogenesis aspects. But after all is said and done, there's still this sort of functional destabilization. And so now we've got our circuit shown here with the excess excitation that develops over time. We know now that this circuit that we sort of counted on in the middle for about 50 years is probably not the whole story. There are also sprouting onto inhibitory neurons. There's sprouting of other neurons that we barely consider to help compensate for the cell loss. And that rapamycin, although it'd be nice if it were effective on just the excitation, isn't, because it's blocking all these other things. And what we have is this excess excitation and excess inhibition that when we block just the new components of it still leaves us with this imbalance. So I'm going to finalize this and just kind of go through a model that I've been sort of thinking about in my head about how this might work. So normally, without the excess excitation in it, what we see is that we've got this balanced circuit with lots of inhibition controlling the granule cells. During epileptogenesis, something happens. A lot of these neurons die, the inhibitory neurons. And part of what I didn't show you is the axons of the surviving cells may actually sprout new terminals and connect with other cells. So you have a situation where you've got fewer inhibitory neurons that are more important and more hyperactivated by the new circuitry doing a bigger job. And so you've got this imbalance where you've got more inhibition, you've got more excitation, and something happens. Something that might trigger a seizure, for example. And even the dropout of a small percentage in that case of these inhibitory interneurons, either by inactivation due to depolarization, or a hypersynchronous activation of all of them that feeds back and synchronizes activity, could actually trigger a seizure or maintain a seizure. And so what we have is a system that's activated both excessive excitation and an apparent attempt, if you want to call it that, to compensate by exciting the inhibitory neurons that effectively destabilizes the entire system. And so with that, I'll end and take some of the questions that you might have. It's just so weird talking to a computer.
Thank you for that great talk, Bret. I had a question to start off. Do you think like a logic between what is the logic between the reorganization? So the granule cells that connect with each other, is it just because they're sort of close to each other in space, or is it because there's sort of some sort of activity dependent way that like a functional reason why that they would start to connect with each other, or is it just because they're so densely packed and they're right next to each other?
That's really a good question. And the way so the way people thought about this and the way I learned it, if that's a way to think of it, it's not really learning because looking back, it's pretty naive, is that a lot of their targets have been killed after the injury or after the status. So a lot of the inhibitory neurons in the hilus, there are mossy cells in the hilus, which are excitatory neurons. A lot of them get killed. And so the sort of idea of conservation of membrane. So you've got an axon going out to a target that disappears. The neurons are apt to replace that target with whatever's nearby. But what guides them up into the intermolecular layer to create these new connections and what's guiding that, I don't know. There are people testing the hypothesis that inhibitory circuitry actually mediates some of that. In fact, I know of two people that are actively pursuing this idea that if you can hyperactivate the remaining inhibitory neurons, you will somehow help guide those axons away from other granule cells. That's all I think it's really early days in those experiments. And so the answer to your question is, I don't know, but it's something we should probably think about in terms of the actual biomechanical features of the brain that allow axons to grow. Probably we need to incorporate some developmental biologists into our conversation. Does that answer your question? Yeah.
Yeah, I'm going to take a question from Bobby. Hey, Bret, great talk. Granule cells and hilar interneurons get a lot of input from outside the hippocampus, such as the entorhinal cortex. What do you think is happening with the long-range inputs to these cells after TBI or after rapamycin treatment? And what do you think is the main effect of all these dentate network changes on downstream regions in CA3 and CA1?
Oh, that's a really good question too. And unfortunately, what we do is we do all our experiments where we've cut off what we think are most of the inputs. And so we haven't done any experiments to actually stimulate those peripheral path inputs, for example, in an intact system. And so we haven't actually gotten to the point where we're looking at larger network effects. I would suspect, based on just studies I've sort of picked up over the years in other areas, that what we're looking at with the axon sprouting and the synaptic reorganization is not limited to, or even primarily, granule cells. I think it probably is something that neurons just do. And so I would expect to find, I would actually hypothesize based on the accumulated evidence from lots of different areas of the brain that this happens to any neurons that are hyperactivated for a period of time, that they sprout axons and form aberrant connections. In terms of the final outcome, again, we're not equipped to do that experiment quite yet. What we sort of are between the extreme local circuit and the whole animal, and what's in between, how those connect with CA1, I don't know, but I did some studies in a different animal model years and years ago that showed that a lot of these same kinds of synaptic reorganization events and cell death and specific kinds of cells occur in CA1. And so whatever is happening in the dentate gyrus, I think we have to assume that it's happening all along the circuit.
Okay. Take another question from Nathan. Dr. Smith, thank you very much for the talk. My question is how ictogenic mechanisms in acute post-traumatic brain injury differ from the ones in late onset epilepsy and TBI, specifically considering that it would be expected that an acute seizure would promote synaptic reorganization and further epileptogenesis, but some studies suggest that acute seizures in humans don't associate with increased risk of developing epilepsy.
So everything you say there is right. An acute seizure does not equate with increased risk if it's one seizure. If it's more than one, there are plenty of people who think that seizures beget seizures, to put your question another way. That's, again, difficult to study, but the sort of the common thought in the epilepsy world is that yes, every additional seizure, especially once the circuit has been reformed, is going to help stabilize that circuit in its abnormal way and maybe make it worse. And so this comes up as a complication whenever you're studying epilepsy and the effects on the circuits, is how much of it is due to the initial insult and how much is due to the recurring seizures that take place months or weeks later. And so the mechanisms of an acute seizure are different, and they're probably an immediate response to whatever the insult is. And once the insult is removed, then that response goes back to normal. But if you have enough seizures, the thought is that eventually the circuit will start to change to make it more likely to have another seizure. And so that's actually one of the sort of overriding philosophies of in epilepsy research in general, is that seizures can beget seizures. But it's true that clinically one seizure does not make you more likely to have another seizure, but two might.
Okay, we'll take another question from Juyun. Thank you for the excellent lecture. Can the models you mentioned explain spreading depolarization?
I think some of them can. So in some of these cases, when you get large numbers of cells firing in synchrony, you can have something that, at least locally, that looks like spreading depolarization. And when people have looked at this in whole animal studies, which is much, you get less detail, but you get more field kind of effects. When a seizure starts, there's often a spreading depolarization that occurs locally, either if it's in the hippocampus or in the dentate gyrus, or in neocortex. And how long that lasts, some people predict that actually might trigger things like sudden death after a seizure if it lasts too long. But they do, if you look at the way the cells are lined up and their geometry, there have been plenty of studies looking at spreading depolarization as an outcome of a seizure, if that answers your question.
And looks like we have another question from Sarah. Have you compared the PTE mice with the non-PTE CCI mice? Could the non-seizure mice potentially demonstrate successful compensatory inhibitory mechanisms? Thanks.
We have some mice that don't develop epilepsy, and in all of our cases, and the same thing with the status models. And what we're finding is that for most of our outcomes that we measure, they're sort of in between. If anything, though, the ones that don't develop epilepsy look more like the controls. And so there might be some things that we could target there. And I understand where you're going with this. And it could be that some of the things that we're really, we're targeting this sort of gross aberrational restructuring that happens in animals that undergo epilepsy, but there could be more subtle aspects that we've sort of just haven't picked up because that's what we're looking at. We do have controls for these, and I'll just tell you in general, they're of all the outcomes measures we look at, they tend to be on the normal side, but not normal, if that makes sense. For some of the things like cell loss, that's not true. Sometimes the cell loss looks kind of the same, but for things, the synaptic reorganization stuff, it's sort of in between.
Got another question from Lee Shapiro. Greetings, Bret. Thanks for the great talk. I'm wondering if you've looked into the CA3 pyramidal cells, are most of the cells forming the excess inhibitory connections, or is it just subpopulations?
We have not, Lee. It's kind of an interesting story that Eric Schnell is sort of taking up, I think, if you look up what he's been doing lately. The concept that the CA3 neurons are involved in this is not that well developed. And I think there probably is some room there to trying to find out which of these neurons are actually participating, but we have not looked at that.
Right, that looks like all the questions in the chat. All right, well, if that's all the questions, unless someone wants to unmute themselves and ask one, I just wanted to thank Bret once again for giving a really nice talk, and thank you to Adam and David for organizing everything. And I think we'll see everyone back next week.
Thanks, everybody.
Thank you.